Can histograms get us out of the years of bad docking? My name is Anna. I'm going to talk about the histogram-based approach for the docking parameters optimization. A fragment-based docking approach was developed specifically to tackle highly flexible ligands, such as single-stranded RNA bound to the protein. It works by splitting such ligand into the overlapping fragments, docking them separately, and assembling back together at the end. The parameters of the energy function, the one that it used to generate docking poses, they have been optimized for the generic case of RNA, and they work poorly for a single-stranded case. It is expressed in the absence of correct poses for a good few fragments, and it can be fixed by optimizing these poses, for example, with our new histogram-based approach. So how does it work? We begin with the energy function and its old docking parameters. For a unique pair of pseudoatom types, we convert its energy curve into the log odds histogram with the help of the Boltzmann factor. Each bar shows the relative probability of occurrence of a distance between pair of pseudoatoms of the defined type in the binding interface. Then we dock one or several RNA-protein complexes with the help of the same docking parameters, and we look at the generated docking poses. For the same pair of pseudoatom types, we calculate the ratio of occurrence of each distance between those pseudoatoms in correct to incorrect poses. Those are the bars of residual histogram. Next, we sum up bars of both histograms together, adjust the energy curve accordingly, and transform it back into the pair of the new docking parameters for this specific pair of pseudoatom types. Repeat for each unique pair of pseudoatom types to obtain the whole set of the new docking parameters. It was the first iteration of the approach. Repeat those steps until convergence, until the residual histograms are flat, to generate an equal distributions of the distances between pseudoatom types in correct to incorrect poses. And that should optimize the number of correct poses by this criteria, by this distance between pseudoatom types criteria. And in turn, that should fix an issue with the absence of correct docking poses. Now let's talk about the results, only preliminary at the moment. So we selected small test and train sets and performed the first step of the optimization for all pseudoatoms of the train set. And then we ranked our docking poses of the test set. Ranking means ordering poses in the increasing order by the approximate energy of interaction. What we want to see is the correspondence between the correctness of the pose and its rank. Most correct poses should have a higher rank. And it was indeed the case for the 2 out of 3 of the test complexes. We can see the correct pose being squished to the higher rank. I want to note that those 2 test complexes, they were pretty similar in structure but not redundant with one of the train complexes, and this can explain such marvelous results only after one iteration of the approach. Now, for the last test complex that looked nothing like any of the train complexes, we don't have any improvement for the majority of its fragments, but for one, we can see actually a shift of correct poses to the higher rank, and one correct pose has actually really high rank. With such promising initial results, now we plan to perform the full-on optimization and publish our results next year in summer or spring. Talking about the perspectives, next step would be to further identify the correct poses by some other criteria with the help of machine learning. That would be all from my side, and feel free to reach out with any questions or comments.